Hello, everybody. It's my pleasure to present our work at the IPPN webinar. My name is Robert Juncker. I'm professor at the University of Marburg in Germany, and I'm also affiliated with the University of Salzburg in Austria. And I will present an ecological perspective on plant phenotyping, more specifically on digital phenotyping of plant individuals and also of plant communities in the field. Trait-based ecology emerged as a very important part of ecological research. And in trait-based ecology, we describe traits, and here we describe the characteristics, the variability, the distribution, and the relative abundance of traits in communities. And these traits or trait-based ecology can link traits with environmental gradients, with niche theory, with community assembly and ecosystem functions. And so it is very central to ecological theory, and because the importance of this trait-based ecology increased over the last years. Finally, colleagues published a book that just came out a few months, the Handbook of Trait-Based Ecology by Francesco De Bello and many colleagues. And so that is just to emphasize how meaningful this part of ecology is for our discipline. And before we think about plant traits and learn about individual studies, we need a definition for functional trait. And that is, according to a definition from 2007, any morphological, physiological, or phenological feature measurable at the individual level from the cell to the whole organism. And that applies to plants. It applies also to animals and other organisms. So that is a very general definition. And how can we use that in ecology when we know about traits? And I will focus on plant traits. So what can we learn from plant traits? Yeah, and so these traits are indicative for species adaptations to the abiotic and biotic environment, and therefore it helps to add a function to our species list that have been recorded in the last century. So that is like the basis for most or many ecological studies, to have species lists. And using the traits, and by knowledge of traits of plants, we can add functions to these species lists. And so traits help defining the functional position of plant species in communities and ecosystems. So in this handbook here is one indication of the importance of trait-based ecology. The other one is this very successful paper on the TRI database by Katke and many colleagues. So the author list is extremely long. And they are compiling trait data from all over the world. So you can see here each of these blue and red dots. That is the data points that are included in the TRI database. And you see the global coverage is quite high. Of course, there are some hotspots, but in general, most parts are covered by this database. And we have many leaf traits, we have many whole plant traits, but also reproductive organs and other parts of plants are considered. And this is already the second edition of this database, and there are numerous studies using these data, because here we can work on biogeography of plants. We can learn about the adaptations of plants on these different biomes and so forth. So they are or this database is a very valuable resource for plant ecology. And this is also shown here in this network of terms that is also part of this paper, how these plant data have been used in the last years. And you see that help to learn about climate change, about diversity, about the functional diversity, which is part of the diversity, ecological and evolutionary studies or questions could be answered, and so forth and so forth. So yeah, this paper appeared 2020 or at the end of 19, and it is already cited more than 200 times. And mostly it's papers that somehow used this data or somehow used other plant trait data. And so in the standard method of phenotyping plants, or to compile this data that end up in this database, is mostly something like that. So we use caliper rules to measure leaves. We use scanners to learn about the leaf area. We use scales to weigh the leaf mass and so forth. So that is the standard method of getting trait data in ecological research. And you see that these simple or rather simple methods, so it's really time consuming, so it's not simple when doing it, but you know, the technical costs are not that high. You can do a lot, and we learned a lot from including traits into our ecological investigations. But I think, and this is what I'd like to propose today, is that the future also in trait-based ecology is this digital phenotyping, and we started last year doing so. Here is the machine we are using, the plant scanner, and I will introduce it later in more detail. I think that is the future, because it is less time consuming and we get a lot of data within, or in a relatively short time. And so that may be an advantage, and let's see what we can do with the data. So in the next 30 minutes, 40 minutes, I will talk about first plant phenotyping in ecological research, and here I will introduce our studies that we conducted more than a year ago, just to show you the potential of the knowledge of traits in ecology and what we can do with it. And later I will continue showing you two studies that we did in the last year on digital plant phenotyping in ecological research. And in both cases, I will give you one example on the phenotyping of plant species or individuals, and also on the phenotyping of plant communities. And to give you a full overview on how we can use this in ecological research. So the first study is on plant individuals. It is Sinabis avensis. That is a study that my former PhD student Jonas Kuppler performed in Salzburg, Austria. Now he's a successful researcher at Ulm University in Germany. What we did here is we had 100 or nearly 100 Sinabis avensis individuals, and we recorded all interactions in the lifetime of these plant individuals, from germination to seed set. And we also recorded the traits, the morphology, the scent emissions of flowers and leaves, and also the phenology. And we also tested the effects of herbivores and flower visitors. Yeah? And we had the question whether these intra-specific variability in traits explained some visitation differences between the plants, and whether this is really plant individual specific, or whether this is just a huge variation within the species, but not very specific to plant individuals. For example, we measured the shape of flowers of these plant species. You see the variation here, and the mean value that we may expect. So that it was just one method. As I said, we phenotyped the plant species in regard of many aspects, morphology, chemistry, and so forth. And now let's look how the flower phenotype may affect the visitation frequency of honeybees, for example, and of solitary bees, in that case, an Andrena species. And what we found is that the size of the inflorescence is positively affecting the number of flower visitors regarding, or the number of honeybees visiting the flowers. For other traits, we found negative relationship, like the height of the inflorescence was negatively correlated with honeybee visitation, but positively with Andrena visitation. And then some other morphological traits like filament length, style length also affected the visitation by bees. So that was a global overview over all of our data. We also translated this into a network. And on the left side here in yellow, you see the Sinabis avensis individuals. This tree is indicating their phenotypic similarity of these individuals. And here are all the flower visitors that we recorded and their interactions with the plant individuals. So such a network may be the result of it is like a snapshot. So you go in the field, visit, observe pollinators, and then, okay, somehow they need to distribute. But the question is, are these individuals, these plant individuals, really unique in their visitation patterns? And one way of testing that is, so we did this several times. So in this flowering season, so maybe 5 to 10 times, we observed the visitors to each of these plant individuals. And then we tested whether the preference of, let's say, of honeybees or of all pollinators remained stable over the time. So a plant individual that was strongly visited in the first observation period, is that one also preferred in a later observation period. And if that remains stable, that is an indication that plant individuals really differ in their interaction partners. And that, of course, has an effect on plant reproduction, the impact niche of the plants, of the plant species as such, but also of plant individuals. So we did this test. I explain you what you see. So on the x-axis, these are just the plant individuals, and they are ranked or ordered by the mean rank over all the observation periods for different aspects. So for example, the total flower visitor number. Yeah, that is here. So this plant individual, the first one, received the lowest number of visitors in all of the observation periods. Yeah, and this one, or these here, they received the most visitors in all of the observation periods. And this means that these preferences are time invariant. So time doesn't play a role. They are also always the most preferred individuals in these populations. We did this for individual taxa specifically, so for syrphids, Aristallini, apis, solitary bees, and so forth. And you see, in most cases, we found significant differences between the individuals in the preference of their of different animals. And this is something we could correlate to the morphology, to the chemical phenotype of the plants. And so the phenotype of plant individuals is really important to mediate these interactions. And you know, pollination is related to plant reproduction, and so also to plant reproduction. And by keeping this diversity in phenotypes in a population, that may help plant individuals to cope with fluctuations in the pollinator composition. And so it helps them to survive in unpredictable environments. So that was an example how plant phenotyping helps us to learn about these interactions in plant species. Now let's have a look at an example of plant communities. That is work done by my former PhD student, Anna-Marie Larue-Kontic. Here we were working in the Austrian Alps along the Großglockner Hochalpenstraße, High Alpine Road. And here we established 8 plots along this elevational gradient. And in each of these plots, we recorded the plant species and we added the functions by adding the traits of the plant species. So we recorded the specific leaf weight, the area, the thickness of leaves. And they are also, they inform us about the adaptation of these plant species in a certain environment. And we have certain expectations for how these traits should change along an elevational gradient. And this is what we tracked in our study. So for each plant community, we recorded the abundance of all the plant species available. And then we measured the traits of these plant species. And by measuring so then we know the functional position of each plant species in a community, and we can then calculate the community-weighted mean, which is the mean trait value weighted by the abundance of the plant species within a community, and we tracked whether this is changing along an elevational gradient. So we did this in an hypervolume approach. In that case, it's 3 dimensions with 3 different plant traits. We have the leaf area, the plant height, and the specific leaf weight. And now you will see these bubbles here. And so each bubble represents 1 plant species. The size of the bubble is proportional to the abundance of these plant species in the community. And you see that is the lowest plant community that we observed with these plant species, and they cluster somehow. And based on this, we can calculate the community-weighted mean indicated by a triangle. And you see that is the mean functional position of the plant species in this area. And now we can add the further communities that we recorded. For example, here the next one, that is the highest one we recorded. And you see it is clearly different from the lowest or from the community in the lowest elevation. So that is clearly a change. But what is with the communities in between? Is that just a result by chance, or is it a concerted shift in traits along this elevational gradient? And yes, it looks like that these plant traits really shift in a predictable manner along this elevational gradient. So the leaf area becomes smaller when we go in higher elevations. The plant height becomes smaller, and the specific leaf weight seems to increase with high elevation, and that is an adaptation to the cold conditions there. So, and also the plant height. So it all makes sense, and that is something that we can confirm using trait-based ecology. Yeah. So here is another overview. You see the rotating coordinate system that helps you to better understand the distribution of the data points. And so the functional composition of these plant communities shifts along an altitudinal or elevational gradient in a concerted way. So, and now the next question is it because the plant species themselves change with an elevational gradient, or is it that individual plant species change along this elevational gradient? Yeah, and we found both is true. So of course we find different plant species in different elevations. But when we found the same species in different elevations, we found that they are also changing their traits as expected by this graph. So let's have a look. So here are 4 plant species that we recorded in at least 2 of these communities. You see the triangles, which are the community-weighted means from the whole communities. And for example, here for Eichemilla fissa, we see the positions of these plants in the different elevations, and you see that is clearly following the trait of the trend of the whole communities. And the same is true for Gentiana, for this and for the last plant species. So we can always see that the plant species themselves also change their phenotype along an elevational gradient, and that is reflected by the community-weighted mean. So it's not only that plant species change with elevation, it's also that plant species themselves change their traits according to the elevation in order to be adapted to these specific conditions that they encounter there. One last comment to that. In another study, we tested whether these traits also have an do not only represent an adaptation of the plant species to the conditions, but whether they also have an effect on important ecosystem functions. And that is another aspect of trait-based ecology, to really learn about the ecosystem functions of plant species or plant communities. And that is like an ongoing PhD thesis of Lisa Maria Ola. And here we measured the erosion of plots, and we recorded the plant communities on these plots and measured the traits of these plots. And we found that plant communities that have larger community-weighted mean leaf area have less erosion on average, and the same is true for those plant communities that have stronger roots, like where the mean root dry mass is higher, then again the erosion is reduced. And so that is showing that these plant traits are also important for ecosystem functions that are in that case also very important for for human interests, because this erosion can be detrimental for our infrastructure. And so another example of why plant phenotyping in ecology is very important. So, and everything I showed you so far has been done mostly using these caliper rules, using like a standard office scanner for the leaf area, sometimes also a scale for the leaf weight. But that is what we mostly did. We went there, used the caliper rules, and measured plant traits. As I said, I guess the future will be digital phenotyping, and so far we did, we were quite happy with the results that we received so far. And the system we are using is a PlantEye from Phenospex, and so I ordered this device 1.5 years ago or 2 years ago, and since then I'm in constant exchange with representatives of this company, and I have to say that the support is really exceptional. So it was a long journey to be there where we are now in order to really be able to generate data. But that was really a great support by the people from Phenospex. So what is the PlantEye? It is this device that you see here. And these slides, by the way, these few slides are provided by Phenospex. So thank you very much for sharing these slides. So it is a 3D scanner. And so it generates a 3D cloud, point cloud. And these points are then like complemented with a multispectral color recording. So we have these 3D point clouds plus multispectral information for each of these points. We have a high resolution, so it's less than a millimeter or there is more than 1 pixel per millimeter, and so that is really a high resolution. It can be used independent from light conditions, and that is an important prerequisite for us to use it in the field. It's heavy duty, another prerequisite for using it in the field, so we don't want to destroy it in the field. It has open data format, so we can use the raw data also. And this is the machine we are using. And that is what it does. So that is an example by Phenospex. So we get 3D images of plant individuals in that case. And there we have these up to 100,000 data points per scan. And each of these data points is, as I said, we get the information on red, green, and blue wavelength, and also on near infrared. And then with the machine, with the PlantEye, there comes a software called Horde Control, and that one is like taking the data and recalculating very useful parameters like morphological parameters, the height, the leaf area, projected leaf area, leaf angle, leaf inclination, digital biomass, and so forth. So, and you see that is quite similar to what we did before. So before we also worked on leaf area, we worked on plant height, we worked on the biomass. And so you see that we can replace our caliper rule with this new machine and get similar data. Additional to these morphological data, we also get physiological parameters, which is the hue, so the color. We can measure the color of the plants, and also like vegetation indices that are related to plant health, the chlorophyll content, the pigmentation, and the nitrogen content. These are proxies, but they can still help us to evaluate the state of the plants. It's the NDVI, the normalized difference vegetation index, the normalized pigment to chlorophyll index, the plant senescence reflectance index, and the greenness. And these are very important parameters, as I said, to learn about different properties of the plants. And the system we have is the MicroScan. And I guess that picture is reflecting quite well what Phenospex had in mind when they were designing this machine. They were hoping for researchers working in a controlled environment in the greenhouse, and then they can scan individual plants there in these controlled conditions. And they don't move the MicroScan that much. We want to take it to the Alps this year, to a very rough area. And so you can imagine that we really need a really sturdy machine that can use in any area that is not like shaky when you touch it. And so we learned from our experience last year that this original design is not really, it hasn't been designed to go to the field really. And so this is not at all blaming Phenospex. They did a great device, but it wasn't really like sturdy enough for us to take it to the field. But it's great that we have the workshop here in Marburg, Gabor Sharenzi and Michael Lizo, and they are really ingenious engineers, and they replaced the whole MicroScan by a self-made frame, and that one is more stable. And we already tried it. It works perfectly, so it does exactly what the original device does. But we are now, so we now dare to take it to the field and do our measurements there. So in the following, I will show you again 2 studies, the one on a plant species level, the other one on a plant community level, to show you how we can use this digital plant phenotyping for our ecological questions. The first one was a bachelor thesis by Helen Phillips, and she was working on Fragaria vesca, the strawberry species. You see one of the scans here rotating, so we nicely get a representation of the plant individuals. And these plants were either well watered or they experienced drought, and some of them experienced windy conditions, as you see here in this small movie. Can I repeat it? Yeah. So, and we had a complete or a full factorial design. So we had plants well watered with wind, well watered without wind, and the same with drought. And then we were interested whether we can use the PlantEye to discriminate between plants that experience the one or the other condition. And so Helen scanned these plant individuals several times, and these are the result. So first, let's have a look at structural traits like the digital biomass on the left side and the leaf angle on the right side. And we see that the biomass was affected. I have to say what you see here. These are the treatments: no drought, no wind with a minus, no drought plus wind, drought and wind, no wind, drought and wind. Yeah, so this is what you see. So the pink box indicates no wind, the blue box indicates yes wind. If the box itself is blue, it is well watered. If it's orange, it's not well watered. And we find that the biomass is affected by drought conditions and by the wind, and also by an interaction of both environmental parameters. Whereas the leaf angle is mostly affected by wind and the interaction of drought and wind, but drought itself doesn't specifically change the leaf angle. So that is what we found here. And when we look at physiological traits like the greenness of the plants and the NDVI of the plants, we found that wind affected the greenness and drought affected the NDVI of the plants. And that means by the number of traits that we are now able to measure just with a single scan, it's 30 seconds per scan, we can clearly discriminate between the effects of different environmental conditions. And that is really helpful, and it's much faster than doing all of it by hand. So yeah, it's really facilitating our research on plant species in that case and how they respond to environmental conditions. So different traits are indicative for different treatments, and this digital phenotyping clearly helps us to assess the adaptations of plants to abiotic conditions. So that is that is a great result, I think. We are really happy with the results of the plant scanner and the digital phenotyping. But I showed you earlier, trait-based ecology is more about plant communities and not that much about plant individuals. And so we wanted to see whether we can scan whole communities and still get meaningful results, although we cannot focus on individual plants or plant species in these scans, but we scan the whole plant community. And can we detect any patterns in there that explain the environment of these plant communities? And that is an ongoing PhD thesis or study by Verena Zieschank. And she really did a great job last year when she was establishing this common garden here. So this is part of the biodiversity exploratories, where she visited 3 regions in Germany. So here, Schorfheide, Chorin, Hainich, and Schwäbische Alb. And she went there and she took grass sods from 1 square meter in 13 areas within these 3 areas. So in total, we collected 39 grass sods of a square meter. And they were then transported to Marburg, where we established this common garden. And here, each of these square meter sod was divided into 4 equal parts. So we have a quarter of a square meter per plot, and they were planted in here. And in the following, they were treated with different land use intensities. So some of them were mown only once a year, others were mown twice a year. Some of the plots received fertilizer and others didn't receive fertilizer. And that again was a full factorial design. So we have all possible combinations of mowing once or twice and fertilization, yes or no. Yeah. And so we established the common garden. Then Verena did these treatments with mowing, with fertilizing. And then from June to November, we regularly scanned the plots in order to get all the information from the communities using the PlantEye. And you know which traits we can get using the PlantEye. And so the big question is, are these traits sufficient to first discriminate between the plots or the origin of the plots? You can remember we got them from 3 different areas in Germany. So are these traits sufficient to discriminate between the origin of the plots? First question. And second, can we use these traits to learn about the treatment? So does the treatments, the mowing and the fertilizing, change the community properties in a way that we can discriminate them? And here are 2 examples of 2 of these subplots. So you see they are quite different. This one is dominated by a single plant with very huge leaves. That one here has many grasses. So you see, we clearly can see the differences in these plant communities. And first, we were interested in how well we can use the PlantEye to estimate the properties of these communities. And one very simple way of doing that is by taking the biomass. So we get the digital biomass from the plant scanner, and we just mowed the communities and then weighted the dry weight of these communities. And we found that this is a very strong and very good correlation. So this is the measured dry weight, how we would do it in the standard way. And this is the digital biomass on the y-axis. And you see that is quite a good estimate of the real biomass. And so we were very happy with this result. And now we know we measure the correct traits here, and maybe we can use it to discriminate the plots. So what you see here is, it's a timeline here on the x-axis. These are different scanning events. Every 3 to 4 weeks, we scanned the plant communities. This is when the plots were established. So shortly after plot establishment, we did the first scan. Then we did the mowing and fertilization. Here we had the second mowing and so forth. Yeah. So this is the timeline. And then we did these scans for all the 169 plots that we had there. And then we used machine learning, random forest, to assign these scans to first the plot origin here in orange, the land use treatment in general, so the combination of mowing and fertilizer, or to the mowing treatment itself or the fertilizer treatment itself. And on the y-axis, you see the accuracy of the machine learning prediction on how well we can predict the plot origin, the land use treatment, or the treatments themselves. And the higher accuracy is, the better we could predict our result. First, let's have a look at the plot origin. You see at the beginning of the season, we were quite successful in predicting the origin of each plot. And this is because that was shortly after establishing the common garden. And so at the beginning, we succeeded quite well in predicting that, because they didn't experience any manipulations by us. And then after mowing, after fertilization, after the second mowing, you see the ability to discriminate this plot was reduced during the season. So these land use treatments that we experimentally applied to the plots was strong enough to induce changes that the plot origin was somehow less and less well predictable. Of course, our treatments were not predictable at the beginning of the season, because here they didn't experience any treatments, but you see that is increasing over time. And so the longer we did these treatments and the longer the plant communities were exposed to these treatments, the better was the predictability. And that is true also here for, so at the beginning we mowed all of the plots, so we couldn't predict that. But as soon as we mowed only half of the plots, the mowing treatment plots, of course we were able to predict this mowing very well, or this treatment. And the same is true for the fertilization. So shortly after we fertilized the plots, we got a very high predictive ability, and that dropped with time after we fertilized the plants. So that is cool. So a very cool result, I think, because now we know we can use these scans to really evaluate the state of these plants, right? And of plant communities, not only plant individuals. And that will be very helpful for future endeavors. And so we just scan the communities. As I said, it's 30 seconds. We get all the parameters about the plant communities, and they are really informative for the properties of communities of interest. But now we know in general we can predict that. But now let's have a look which properties or which PlantEye parameters are informative for which treatment, mowing and fertilizer treatment. And here we found a very nice difference between these two treatments. Of course, the mowing treatment, here we had the number of voxels, so we translated the point clouds into voxels, and the number of voxels is the number of voxels that contain information in the scans, the digital biomass, the light penetration depth, the hue, and the height. Of course, these structural traits, they inform about whether the plot was mown or not. That is a little bit something we expected. Yeah. So, but still we can measure it with a PlantEye. That's cool. We can see the results. The fertilizer treatment, however, was best predicted by the physiological traits measured by the PlantEye. And so depending on which treatment we are interested in, we can use different parameters of the PlantEye, again, showing the potential of this digital phenotyping device, because now we can assess different properties of plant communities, and we can really discriminate them by the choice of parameters. So the summary of this part is, first, plant communities respond rapidly to land use changes because the accuracy in predicting them, the changes, increased very shortly after we did the treatment. And what we can say after this first season of using it, digital phenotyping is a fast and reliable tool to evaluate the state of whole plant communities. And maybe you remember the definition of a plant trait or of a functional trait that was measurable at the plant individual level. So in a strict sense, we are not measuring functional traits because we don't measure them on the plant individual level, but we measure them here on the community level, but still we can learn a lot from that. So to conclude, everything I said before is trait-based ecology really advanced the field of plant and community ecology. And why? Because it is adding functions to species inventories. And by the use of digital phenotyping in the field with our redesigned MicroScan from Phenospex, first, this will reduce the workload associated with trait measurements. So my students spent countless hours to measure these traits by hand. And maybe we can reduce this workload by just using the scanner. I don't think it will completely replace this manual measurements, but I think it will clearly be a complementary approach. And I guess often we will now not do the manual measurements because we are happy with what we get with a digital phenotyping. We can add further meaningful traits that are not accessible by our standard approaches. So the NDVI, for example, is something we couldn't measure in the last years, but now we can, and we saw that this is indicative for wind, it is informative for fertilization and stuff like that. So this really adds to our knowledge. And I think that is also something that we are developing currently. We also can get the raw data of the scans, like the 3 dimensions of the X, Y, Z of the points in the point clouds, plus these 4 spectral informations like red, green, blue, near infrared. And so we have a data frame of 7 columns per scan, and all the 160,000 or something like that data points. And this raw data can be used for artificial intelligence, for machine learning approaches to really allow for more detailed insights, for feature extraction of these scans, and for, we hope, predicting diversity in future. So let's see how we can proceed with that. And I think the digital phenotyping of communities closes the gap between the classical trait-based approaches, where we measure individual plants, and remote sensing, where we have much higher resolution, a much smaller or lower resolution than we get with the PlantEye. And I think this is closing the gap, and all the information that is within this gap is now finally accessible to community ecology, to trait-based ecology. And so I'm very happy with this device and with all the potential studies that will follow in the future. So thank you very much for the invitation to give a talk in this webinar series. Thank you also for listening and yeah, watching the webinar. And yeah, if you have questions, contact me in the session or send me an email. In any case, yeah, thank you very much for listening.